I must admit, when I saw the title of this next talk, I was mystified and quite intrigued by what you do by putting an imaging device on a computer mouse. I mean, what's the point of that? But it actually turns out it's got nothing to do with that at all. This talk is from the life sciences. The point of the technology is to study the function of brains of mice. And Shung Chieh from Inscopix is going to come up and tell us all about that. And let me...
Thanks. Yeah, the title is a little bit of a red herring. Actually, the talk is more about neuroscience and using embedded technology, or embedded vision in particular, to learn more about real vision or biological vision. So a little bit about Inscopix. We were founded around a small wearable, as you see here, sitting on top of a mouse. It's a single photon epifluorescent microscope, which is used to watch fluorescence. And the fluorescence we're seeing is really due to this green fluorescent protein, which is selectively introduced into certain neurons in the brain. And in this case, what you're observing is the hippocampus, and the cells that are lighting up are really hippocampus place cells. And these cells actually encode what the animal is doing in the middle of this arena. And in this particular case, if you actually decode the activity of these flashing neurons, you can actually come up with a very good prediction of where the mouse is in the middle of the arena. So we spun out of Stanford about 5 years ago, based around this technology, and we've already been shipping this microscope and basically a whole workflow for understanding brain circuits. We've been named as a industrial partner of the Brain Initiative, and we were also cited as a World Economic Forum tech pioneer. We have over 200 labs using this microscope, including several Nobel Prize winners. And we've really been driving a lot of neuroscience understanding of function and dysfunction, all sorts of behavior, all sorts of mechanistic understanding of how the brain works. So the next slide shows two specific examples of the type of neuroscience that we're supporting. The one on the right, I'll start with the one on the right. This is a study of an epileptic mouse model. And what you'll see is that in the bottom graph, you'll see a graph which will show both the an electrode behavior, the behavior of an electrode that's been implanted in the same area, and then the calcium activity as a seizure is about to occur. And what you'll see is that it's preceded by this very interesting wave-like phenomenon that goes across the field of view, and then you actually see the seizure occur. And there are all sorts of phenomena that are observable with this technique, and as you can see, it's a very complicated, how exactly that the brain works. On the left, what you see is basically an observation of hunger in action, and the before and after videos. Let me play this. The before video shows some AGRP neurons in the hypothalamus, and these are known to be kind of, I would say, signaling of hunger. And the after picture is the behavior of these neurons after injecting what's known as a hunger hormone, ghrelin. And by using this technique, neuroscientists have been able to kind of tease into the causes and then the actual manifestations of behavior based off of the circuit behavior. So we're not just about mice. We've been slowly extending our application to other animal models. We started in mice, but we've already been used on rats, and we've recently started to work on non-human primates. The whole goal of this is to generate findings which are more and more relevant to humans, and then hopefully lead to better understanding of human neural activity and behavior. The other, in terms of this audience, I'll focus more on the system engineering and some of the challenges that we faced, as well as some of the opportunities to leverage a lot of the great technology that's been developed in embedded vision. In the first place, we've been, with the start of this project, we were focused on the microscope, but we've moved upstream in terms of really facilitating acquisition and integration of all sorts of data. And then the second place that we've spent quite a bit of effort on is in the analysis of this data. But first, let me describe some of the extreme challenges that we face in designing our system. The first one is the fact that this microscope has to be borne by the mouse, and all sorts of things that might be tolerable by a human aren't tolerated by a mouse. The first one is form factor. So the microscope design is actually very straightforward. It consists of essentially a grin lens, which is coupled to another grin lens, which is inserted in the brain. The grin lens that's inserted in the brain relays the image from deep within the brain to the surface, and then that is passed through the optical system. We have to illuminate the field of view, and in this case, we're showing our second generation microscope, which has 2 LEDs, and I'll describe the purpose of those LEDs in a second. But essentially, the illumination is passed selectively down into the field of view, and then we pass the fluorescence image straight through the dichroic mirror and then up to an image sensor, which is at the top of the microscope. Now, the reason that we have 2 LEDs is because one of these LEDs is specifically for exciting the fluorescence that we're observing in the brain. And then the second LED actually speaks to a new technology that we've been working on, which is really coupling optogenetics with this calcium imaging modality. And the purpose of the second LED is really, there's now a way of introducing another type of protein, which is known as an opsin. This protein is photosensitive, and when you shine light on it, you can either inhibit or you can excite a neuron to fire or not fire. And the engineers euphemistically refer to this LED as the mind control LED. And we refer to the other LED as the mind reading LED. So what we really have here is a brain-machine interface, if you will, and a whole system that's starting to revolve around it. But in terms of the form factor, it's really pretty constrained. We have to fit on top of a mouse, not be so heavy that we're affecting the behavior of the mouse, because the whole idea of this is to be able to observe behavior. And that really constrains all the things that we can do. So things like wireless, cable, all these things, we've had to carefully engineer this so that it's compatible with the mouse application. Our roadmap is actually working on shrinking this microscope, because we would love to put a few more of these into the brain. Right now we can see about 1,000 neurons in about a millimeter field of view, but that doesn't really tell you that much after a certain point. It certainly tells you a lot more than the technology that has existed to date, which have really consisted of electrodes sticking into regions of interest and not that many electrodes to begin with. So the state-of-the-art ECOG arrays or EFIS type arrays, they stick about 100 or a couple hundred type of electrodes into the brain, whereas our optical technique really allows you to see 1,000 neurons and then be able to go up and down within the region of interest. So one of the ways that we've been trying to target, shrinking the microscope is really, just picking smaller components. And then after a certain while, what we found is that the optics is very challenging, because soon you run out of the fact that you're limited by the index of refraction, and then refractive index doesn't work so well. So we've had to introduce some diffractive optic elements, and then we've also introduced some fiber type, borrowed some concept from fiber coupled optics and illumination. So these are some of the ideas that we're pursuing on that front. The second major challenge, of course, is that our wearable has to go on top of a mouse and is intimately connected with a pretty crucial organ, the brain. And the first thing, of course, is that we can't heat up the brain. It turns out, of course, as you know, that the brain really likes this very narrow temperature range, and if you increase it even by a degree centigrade, that's pretty harmful to operation. So in the one case where we introduced the optogenetic stimulation LED, it turns out that that biological protein needs quite a bit of power to actually excite it. And that's different than the excitation LED or the fluorescence case. And so we've done quite a bit of checking of that LED operation to make sure we're not heating up the brain. And what you'll see is that we tried a whole bunch of protocols for running that LED. And it turns out that because the biological time constant's a lot shorter than the physical, the ability to pulse modulate this light, we're able to kind of stay within that narrow band. The other challenge, of course, is how do we check that we're not really heating up the brain? It turns out, of course, that when you're dealing with these small tissue samples and then putting another sample, the mere presence of a thermocouple changes the thermal environment. And so you're affecting the environment that you're trying to measure. So we've also coupled a lot of careful characterization using thermocouples and actual biological samples, checking them as well with other means with simulation. So it turns out that there has been this paper out there that has created a Monte Carlo simulation of the light and the temperature rise induced by optogenetic light stimulation. We actually downloaded the code and then ran it on our situation, compared it against actual thermocouple measurements, and then dialed the whole thing in. So we're pretty confident that we're not overheating the brain. The third challenge really actually echoes a lot of the things that we heard about in general on deep neural networks and embedded vision in general. And I'll just take a step back and say that what we found is that we're only looking at about 1,000 neurons in a small field of view, and we're trying to pair that with behavior. And so we're actually doing the reverse problem of a lot of the things that the deep learning world is trying to do. We're trying to decode what the brain is doing based on all the observations that we see. So what we found is that we are missing a lot of information, a lot of contextual information. And what we also have found is that we need some edge computing in order to be able to lead to better insights on what's going on in the brain. So we've been developing what we've called the Inscopix hub, and it's really built around an NVIDIA platform. And the whole idea is that it centralizes all the data collection from a multimodal experiment where we're combining our optical technique with EFIS techniques with other techniques. And as you'll see, as you saw in the first video, it's very, the types of papers and types of talks that are given really pair all sorts of data together. And what we found is that there isn't really something integrating all this neuroscience data very elegantly. And there are a lot of postdocs and grad students out there manually working with data, trying to align pieces of data and then trying to get it all to sync up. So we've decided to pursue a hardware approach to simplify all that. So what we've done is with that platform, we're having it be the kind of the nerve center, if you will, of all the data that's coming in. And the whole intent here is to be able to timestamp that data and then eventually build in metadata extraction capabilities so that we can compress the information, if you will, rather than just the raw video. And then the other thing that is also has been a side benefit from developing this hardware layer in the middle is the fact that before we had a camera that was plugging using USB connection directly into a client PC, but we found that there's so many client PCs with so many different different software on them that it was a really kind of a support nightmare. And so by controlling this middle layer of software and then controlling the interface back into it, we've been able to really minimize a lot of the support issues that we had to deal with in the past. We've adopted a framework. The software framework really is nothing new, but should be very familiar to those that work in the IoT space or even this edge and cloud computing model, which is really built around an event bus and then a channel for pushing high volume data or high bitrate data, which is really the videos, both the behavioral video as well as the calcium imaging data. And then, of course, once we get all of this, these nice videos, we need to turn it into something which the neuroscientists really understand. And they're not used to dealing with the videos, but they are used to dealing with cell traces, which represent neural activity. And so this example, let me just play it, shows a mouse in a linear track. And what you'll see is that as this mouse turns around, comes back, there's one neuron in particular that fires. And that neuron really represents one aspect of location and in some sense, velocity. And so we started out with very classical computer vision type techniques for trying to extract out behavior or extract out correlations between calcium imaging and the actual behavior. We hope to build upon that with machine learning techniques. We haven't done this yet, but we've been developing both the hardware and the software with this end in mind. So the data analysis is complicated, and this comes back to the first challenge, which was the form factor challenge. The fact is that because we were constrained by size, we've had to rely on some optics which have a lot of distortion or other non-idealities. So, and then the other thing which is noticeable in hindsight, but not necessarily when you start out, is that when the mouse is moving around, the brain is also moving around too. So we actually have this complicated brain motion that's moving on top of the fact that these neurons are flashing. So we're currently using classical computer vision techniques to try to do motion correction and then background subtraction. And then we hope then to use machine learning techniques to then start to pick out these neurons and their activity. So the current, in that activity, the one thing that we ran into is the fact that ground truth for such training or such a situation is very hard to tell, because we don't really know what the brain should be doing. We're trying to figure out what the brain is actually doing. So we've also been doing a lot of simulation of purported cell activity and then generating simulated movies with the intent of using these to train a machine learning algorithm. So we're right in the middle of that, and hopefully that'll all work out appropriately for us. So to summarize, it's really exciting to be at Inscopix and to be in the middle of all of this. We're at the confluence of genetic engineering, biological, new biochemistry, new molecular engineering techniques to really tease apart what the brain is doing. And then to be able to couple that with an embedded vision system is really exciting. It's satisfying to hopefully participate in the loop where science will illuminate learning techniques and then inform future technology development. So I'll just close by saying that, we make a pretty simple system, but it's challenging because it's got to go in a mouse. It's a very typical microscope, except that we've got to make it weigh less than 2 grams. And then it can't burn enough power. It can't burn so much power that it's heating up the animal. So form factor, power, and thermal challenges are very acute. The data analysis is also challenged by that because we have a lot more optical distortion and these kinds of things which we have to back out of the system. And then really the two challenges before us and really and opportunities revolve around integrating all the pieces of data that are necessary to really tease apart what the brain's doing and then to make sense of it all. So I'll conclude by thanking our team. We have a team of both neuroscientists, engineers, software and hardware, and it's been a wonderful group to work with, and certainly appreciate coming here to the Embedded Vision Summit, and appreciate being included in the program. It's really enjoyable to see the confluence of all these technologies and then be able to apply it in this way. So.
Well, thanks very much. That truly is fascinating. That's a big departure from everything else that we've seen today. We have plenty of time for questions if anyone wants to come and ask. Step to the microphone, which is in the, I can see one taker already.
Okay, so you touched on the sort of feedback loop of the mind control LED, but then you didn't talk about it anymore.
Yeah, I didn't.
Just talk about that for like
Okay,
15 minutes.
So I didn't talk about that mostly because it is a tricky subject for us. But yes, we're very much interested in tying the read to the write and then doing it in a closed loop fashion. The one video that I showed about the epileptic mouse model is really kind of a precursor to this. So for example, as you're watching the calcium signal, and this is before a seizure occurs, you can envision that we could stimulate the right neurons in the right way at the right time and be able to prevent a seizure. But we haven't gotten there yet. That's certainly something that we have in our sights.
Okay, well, if there are no more questions, I'll thank our speaker again. Thank you very much.